Hey everybody, and welcome to Thought Leader Thursday. I'm Beth Usry, the Director of Customer Engagement here at Quality Executive Partners, and today I'm going to be your moderator. This webinar series is brought to you by Quality Executive Partners. And for those of you who aren't familiar with QXP, we are both a consulting and a technology firm that provides innovative solutions for the life sciences industries. In 2019, we launched Virtuosi, and it is literally transforming learning and education in the pharmaceutical industry. Our first series combines video and virtual reality interactive experiences in an 80-hour curriculum focused on aseptic manufacturing and microbiology. I'm so excited to lead this webinar and introduce you to our two experts that are going to be joining me today. You may know these two folks from consulting projects at your site, conferences, or training courses from industry organizations. But first, let's bring out Bob Farrer. Bob is the Vice President of Consulting Operations here at QXP. He holds a BS in Engineering Chemistry from Stony Brook University. He also has over 30 years of experience in industry and is active in both ISPE and PDA. Bob has presented courses for both PDA, ISPE, as well as other trade industry organizations.
Hey Beth, how's it going? For a minute there, I thought I got a promotion I didn't know about.
I know, I was trying to promote you up. It's so great to see you, Bob. I'm so glad you're here with us today. Yeah, for those of you who didn't see Thought Leader Thursday earlier in the year, in this we had a webinar series. There were 6 of them earlier this year. Bob, why don't you share a little bit about your background and what inspires you to teach?
Yeah, sure. So, when I first started in the industry, as you said, over 30 years ago, I worked with people who actually designed and developed the processes of the products that we were manufacturing. So I was able to get a real deep understanding of the science of what we were doing, right? So, and because I had that fundamental understanding and that knowledge, it was powerful because it really helped propel me through successes in my career early on. And now what I enjoy doing is I enjoy taking that concept and bringing it now to new people. So I try and mentor, educate, and lead people and help transfer that knowledge so that they can understand the science behind the compliance of what they're doing, ultimately with the goal of trying to reduce human error in pharmaceutical manufacturing.
Yeah, thanks for that, Bob. Having been the recipient of your mentorship and your teaching, I can attest that you are a thought leader in those areas and certainly have a passion for it. So we appreciate that. So let's bring out our second thought leader, Becky Brewer, who is the Vice President of Strategic Practices. Becky holds a degree from Mount Holyoke College. She has more than 30 years of experience in industry, is a member of PDA Scientific Advisory Board, and is the group leader for the PDA Aseptic Processing Interest Group. Becky is also a member of the newly sanctioned Advanced Therapy Advisory Board and is a PDA laboratory course instructor and also a classroom instructor for several courses with ISPE. Hey, Becky.
Hi, Beth. Hi, Bob. Glad to be here today. Thanks for the wave, Bob.
So great to see you, Becky. You're a prolific industry contributor. But for those who don't already know you, can you share a little bit about your background and what motivates you about this industry and why you like to teach?
Sure. I started my career in aseptic processing and had a real love for microbiology, which, as you know, is a really challenging field, and it is, really puts us to the test when we find environmental isolates in our facility. Today's topic, which combines an activity that we have to do to control our environment, the facility cleaning and disinfection, combines my love of both aseptic and micro, and so I really look forward to talking about that today.
Thanks, Becky. And the folks that we have on the call today, Becky and Bob, you guys are really making a difference in life sciences through your experiences, training, your passion, through the mentorship and through the teaching that you folks do on your consulting projects and in the industry organizations that you're associated with. And this is exactly why we started Thought Leader Thursdays and giving the audience and giving you all access to folks like Becky and Bob. And it's important for us to share the knowledge and to transfer it to folks who are in the industry every day.
Mm-hmm.
So now that we've gotten to know our thought leaders, we'd like to get to know the audience a little bit better. We would like to understand what your responsibilities are relative to facility cleaning and disinfection. So We're going to do a polling question here, and it's actually popped up already. This is going to be one of 2 polling questions. Consider this one practice, and the next one you'll know exactly what to do. So if you could see it up on your screen, the question here is, my role in facility cleaning and disinfection is. So you can go ahead and answer that question. This is completely anonymous, so I can't even see who's answered. So let's take a look. I see the votes are coming in. Excellent. And if you aren't able to see it on your screen, it may have switched modes. So take a look around. You should pull that up. All right, we've got some great responses. We'll keep it open for a few more seconds here. If you haven't already voted, go ahead. And I see it's slowing down. I think we've got most everybody in. So why don't we go ahead and close the poll and see what kind of response we got.
Wow, we've got a little bit of everything in the class, and it looks like the majority are saying that they're in more than one of the areas, both definition and implementation, as well as monitoring or auditing the program. So that means that we've got an excellent program that's well targeted to the audience that we have. So that's good news.
Yeah.
That's great. I should also mention that one of the very best parts about the format that we hold these webinars in here at QXP is getting the audience an opportunity to ask questions and getting the audience and participants involved. So you'll be able to ask questions at any time throughout this webinar, and at the end of the session, we'll be doing question and answers with Bob and Becky. So in order to ask a question, you just go to the control panel in the upper right-hand corner of your screen. You're going to click on the orange arrow to expand the menu box. Select the questions box, and you can go ahead and type your questions in and submit them there. We look forward to seeing all of your questions. Again, you can ask questions all throughout the webinar, and we will gather them together for the end of the session and do a question and answer session. All right, so why don't we just get right into today's webinar topic, which is facility cleaning and disinfection. Today, we're going to get a chance to see educational videos that help us understand the importance of cleaning and disinfection practices and how to determine cleaning effectiveness. You'll also get to see a demonstration showing proper techniques in the facility cleaning in our virtual laboratory environment, which is really cool. I think you guys are going to really enjoy that. And please note that as we cue in and out of the videos, you may have a short delay or the screen may change color, but just hold tight. Your screen will come back. It's just what happens when we switch back and forth between videos. If you are on a mobile device, you may need to tap or swipe the screen as the video loads, and you may need to adjust your volume. So before we get focused on how and what of facility cleaning, let's talk about why it's so important that for our cleaning and disinfection processes to be robust and effective.
I think I'll take that, Beth. So it's really an important element associated with our sterility assurance program, right? Cleaning and disinfecting the environment is the foundation to ensuring that we have a satisfactory environment in which we can produce our products. So it's so important to plan this and test the techniques that we're intending to use and to ensure that they're really a robust method for ensuring that we keep low levels of contamination in our facility. You know, in this day and age, it's not enough to just test our end product and say our end product isn't contaminated. It's really about proving these building blocks, and the facility is certainly that foundation. We really don't want to put anything into our product that could harm patients, and so it's important we start right from that foundation.
Yeah, and Becky, just one more thing to add to that. You know, everyone needs to understand that a robust cleaning and disinfection program relies on more than just the people who do the work, right, and more than people who plan the work or monitor the environment afterwards. It's really critical that we have a multidisciplinary functional group, you know, from operations, engineering, quality assurance operations, even purchasing has a part in this whole thing.
Well, I hadn't really considered all of the different functions within the organization that would be involved in defining the cleaning program, particularly purchasing. But you know, now that I think about it, I can see how each one of those departments would be involved. So I know that critical processes in manufacturing require qualification, right? So I assume cleaning and disinfection processes need to be qualified as well. So what does cleaning and disinfection qualification look like?
Well, it usually takes the form of 3 steps. First, we're going to determine which agents we want to use, and that's usually done by a literature search. What we want to do there is we really want to understand the chemical activity of potential disinfectants based upon the material surfaces that we have in our facility or our plant to have in our facility. There's a lot of common materials, stainless steel, glass, Lexan or plexiglass, but then you get into some of the other materials that may be specific to your process that you really need to understand whether these disinfectants are going to have some sort of a corrosive or an interactive nature with those parts. So that would be the first part. The second part is to do a laboratory study. So this would be a disinfectant efficacy study, right? Where you take a coupon of pieces of material throughout the facility, you inoculate it with bacteria using both environmental isolates as well as a panel of organisms recommended by USP or EP, and then you determine the concentration and the contact time that's necessary to get the required log reduction on these coupons in that laboratory study.
Right. And then, Bob, we often do a field study or an in situ study where we take that experimental confirmation of the effectiveness of that agent and really prove it in the environment with real surfaces and real materials. And this gives us an opportunity to check our technique and ensure that our technique is for applying that solution is going to be effective for covering all surfaces and ensuring the attributes of the process that we determine, including things like the wet contact time.
And the people, right? The chance to test to verify that people are able to do the procedure properly.
And their training, absolutely.
Yes, yeah.
That's great. There's definitely a lot involved. Is there anything else that needs to be considered for routine cleaning?
Yeah, I think the most important thing for routine cleaning is the plan for how we're going to attack the facility. You know, our facilities are very complicated, and I think the first video that we have today talks a little bit about establishing the plan and making sure that we've got a good approach for tackling complex surfaces.
All right, great. Let's go ahead and queue up the video and take a look.
Now that we've completed all the various qualification activities, we're ready to start disinfecting on a routine basis. However, we need a plan detailing how we're going to physically disinfect each individual room. The first thing is to ensure that the area is actually ready for sanitization activities. All manufacturing activities should be completed, and all batch components, materials, and change parts should be removed. Then, a line clearance review should be performed by the responsible department at your facility. To remove product residues, parts that are in contact with product and have become soiled can be removed and subject to clean out of place or COP. Alternatively, they can be subjected to clean in place or CIP. As part of the room cleaning plan, all non-stationary carts and equipment, such as environmental monitoring equipment, should be removed from the room. These carts, including the wheels and equipment, should be sanitized according to procedures prior to relocating to the designated storage location. Upon approval, the sanitization activities should be started with a treatment of the production equipment surfaces. These should be sanitized using approved sanitizers and lint-free wipes. The cleanest areas of the facility should be disinfected first, unless they are physically segregated from other areas. In other words, ISO 5 areas should be disinfected before ISO 7 areas. The area should be disinfected from the top to the bottom. Ceilings should be treated before walls. Walls should be treated from top to bottom before floors, and floors should be treated from the furthest area away from the door outward. These patterns ensure that the operator doesn't walk on freshly disinfected wet floors. This process continues until the operators have cleaned out of the room or zone in which the cleaning was occurring.
Later on in the segment.
Well, there certainly is a lot to consider when we're talking about cleaning and disinfecting. So once we have a cleaning plan defined, let's say we've defined what that's going to look like, I'm guessing that there's a specific method to the cleaning that needs to be followed.
Yeah, how about if I take this one, Becky? So Beth, in the interest of brevity, when I selected that segment, I didn't want it to go on too long, but later on in the segment, we actually talk about having a floor map, literally, a diagram to understand where I'm going to clean first, especially around large fixed equipment. So imagine you've got a number of bioreactors in a room or tanks, things that can't be moved. I need to have a plan to actually go in and make sure that I'm sanitizing things. It's not just as simple as saying, clean the surface, clean the wall, clean the floor. It's how am I going to do that? And part of that plan is am I using a mop? Am I using a wipe? So when we're disinfecting, the mechanical action of actually putting a disinfectant on a surface and then physically removing it aids in the removal and kill of microorganisms. So I might use a mop on large flat surfaces. I'll need a wipe on irregular surfaces or where I have to get on crevices, let's say a door handle or a window around a door frame. So those are some important things to consider. And then for surfaces that aren't or are large and let's say lend themselves to spraying, we might consider using a sprayer, right, to saturate the area. And then I would use the mechanical action of the mop or the wipe to remove that after a certain dwell time sits there. I might even use buckets, as we can see in the image on the screen, a 2-bucket system or a 3-bucket system where I'm using disinfectant to apply and then I'm wringing out the disinfectant afterwards. It really depends upon the facility and the area that I'm trying to clean, how I'm going to develop that cleaning plan.
Yeah, and in addition, Bob, to having that plan and identifying the tools, we also need to pay attention to our technique, right?
Mm-hmm.
So we've got to make sure we're using overlapping strokes, that we're going unidirectionally. Usually we do top down, as talked about in the clip that we just watched, because we want to ensure that we're not recontaminating any surface that we already cleaned. And as we talked about, we go from the back corner to the door. So there's lots of technique that we need to perform relative to that space. This is one of the things that the VR and Virtuosi focuses on. And I think we have a clip coming up, Beth, where we actually let somebody practice working on the mopping of the floor in a room. Now, in the VR activity, there's spraying activities and mopping activities, there's wall activities. The segment we're going to watch is just the floor. And so we're going to focus on the floor in this. Now, the view when we capture it by camera is a little bit smaller than what someone would see if they were wearing the VR headset themselves. In the VR headset, you have peripheral vision, and it's just like being in this room. You can see all around you, right? But when we capture it on film, we have to take a step back and film from a little bit farther away so that that screen capture captures a little bit more of the image because we don't have the benefit of being in the headset. But let's take a look at it and see what that VR clip looks like.
All right, yeah, thanks, Becky, for describing what we might see there. Having been in VR before, I know that you can turn around and look up and down, and I'm glad you described what specifically we're going to see in this clip. And just a reminder, you may have to adjust your volume for the videos. So if you're not hearing sound, it may be because you need to adjust your volume. So why don't we go ahead and get it started and we'll take a look.
Okay, great.
As we begin this video clip, we see on the wall a poster with images depicting 2 possible patterns that can be followed when mopping the floor virtually in Virtuosi. Now, in the real world, your organization may have other options for floor mopping patterns, but in Virtuosi, we are only cleaning a very small area so that we can demonstrate how to maintain a constant leading edge on the mop to achieve a unidirectional overlapping stroke. So let's watch as the learner stands in an empty room with just a set of goggles and a controller in each hand. They focus their attention on the virtual instruction board in front of them and point at the continue button when they are ready to begin. The immersive starts with an introduction and a set of instructions.
The cleaning and disinfection of floors in classified environments always presents a number of challenges. First, portable equipment may be within the space that needs to be removed from the room in preparation for cleaning. Second, fixed equipment typically stands on the floor, so whether you are faced with flush-mounted skirting at the bottom of the equipment, which is ideal, or legs that the equipment stands on, which will require cleaning beneath the equipment, a plan is needed for how the cleaning will be conducted. A large mop head may not always be suitable for ensuring adequate contact on irregular surfaces. It is sometimes necessary to use a sterile cleanroom wipe saturated with disinfectant to appropriately disinfect around irregular surfaces, such as legs of equipment on the floor. However, the clean room wipe should be applied with the mop or other tool, not with your gloved hand. Remember, good aseptic technique requires that we never directly contact materials that are on the floor. Finally, the room may have multiple doors. Therefore, the sequence of cleaning will need to be carefully planned. A central principle of disinfecting a floor is to ensure that you never walk over or move equipment over any section of the floor that has just been disinfected. To facilitate that, there are some basic mopping sequences that are commonly observed. Let's review those sequences first, and then discuss some other considerations. If there is equipment or other obstruction on that path, trace around that equipment with the mop to ensure coverage. The leading edge of the mop should be consistently maintained in all strokes.
Here we see some visual cues Virtuosi provides us, such as the mop saturation indicator, letting us know when it is time to rinse and wring out the mop. Finally, notice that if the learner needs to refresh themselves on the mopping pattern, a quick view of the poster on the wall reminds us of the final path to take.
So I never really considered how important things like mopping the floor are, and it's really gotten me thinking about the way that I do things when I clean my house, and that I'm certainly not being as efficient as I could be. But seeing why good technique is important, that was a great visual for why it's important. It makes you really think about how you're doing what you're doing. So if we have a sound cleaning plan, and I'm following this through on what you all have been teaching us here, is that if you've got a sound cleaning plan and we use good technique, then we can be sure that the cleaning and disinfecting is working then, right?
No, not exactly, Beth. We know that it should be working because we've got the laboratory studies. So we know that it should be working, but we still want to ensure this, and we're going to do that through our environmental monitoring program.
Right, and that starts with an EMPQ that precedes that, where we do a performance qualification for our environmental monitoring to ensure that the actual recovery that we see in an area meets our expectations. And so this really challenges the performance of the cleaning and disinfection of the facility to ensure that our process is working effectively and controlling things as we expect.
Yeah, and we also have to consider the frequency of our cleaning. We have one more clip from Virtuosi that covers that topic. Why don't we take a look at that, Beth?
Okay, yeah, that sounds great. Let's go ahead and cue it up, and we'll take a look.
The frequency of effective facility disinfection is an important decision. Cleaning that is too frequent impacts the ability to manufacture, consumes more materials and supplies, and could cause premature corrosion of the facility and equipment. On the other hand, infrequent disinfection could contribute to a buildup of microorganisms that will put product and therefore the patient at risk. A company must decide how best to establish the initial disinfection frequency. Ideally, this would be established based on data collected during the PQ. For example, disinfection frequency could be based upon area classification. This might take the form of a schedule requiring ISO 5 areas to be disinfected daily, ISO 7 areas to be disinfected weekly, and ISO 8 areas to be disinfected monthly. Conversely, a company could base disinfection frequency on environmental monitoring data. However, that data is not available in real time, so any disinfecting activities are performed in response to data that is 3 to 5 days old. This model would also disinfect more frequently due to normal shifting of microbiological flora over the course of different seasons. When used alone, this approach makes it difficult to plan around the disinfection activities. Most companies employ a combination of these two approaches, with a classification-based frequency that takes into consideration the risk inherent in the processing methods. This is then augmented based upon real-time results as part of a documented risk assessment.
Well, there's just so much to think about and consider when you're defining a cleaning and disinfection process in a program. So as I said, one of the greatest things about the format that we use here at QXP for our webinars is audience participation. And, a lot of us have only had experience maybe at 1 or 2 organizations. And by polling the audience, which is what we're about to do, on different questions and topics gets us a really good understanding of a broad understanding of how others are doing these activities within their own corporations. So why don't we go ahead and do another poll? Keep in mind that this is anonymous. You may have to switch from full screen when you're watching the video to see this poll question. And it's, how does your facility define cleaning frequency? So go ahead and answer that, and we'll get a beat on how other companies are defining cleaning frequency. We've got a bunch of responding folks here. We'll give it just a few more seconds. So how does your facility define cleaning frequency? And we've got more people responding. We'll keep it open for a couple more seconds here.
It's kind of like popcorn, Beth, right? As you see the responses coming in, you're listening
Yeah,
to
it's like,
the
ooh,
clock slowing down. Yeah.
All right, well, I think that we can wrap this up. I think everybody has responded. Let's see what we got.
Yeah, that's about what I think we expected. As the video stated, most times it's a combination of techniques where we base it on the zone classification initially, and then as we continue to gain data, we use that data to inform whether that frequency needs to be increased based on specific incidents in that environment. So again, I think that's very consistent with what we expected.
Okay, that's great. So in the spirit of continuing to keep everybody engaged and audience participation, why don't we open it up and start our Q&A session? So if you have not asked a question and you would like to, you can go ahead and submit that question anytime. We are fielding those questions. We'll go ahead and get this started. And let's see here. So here we go. When the regulations talk about rotation of disinfectants, what does that mean? Oh, does that mean that I have to switch between two similar disinfectants?
Yeah, I'll take that one, Becky. No, it really doesn't mean that you switch between two disinfectants necessarily. And I want to qualify that a little bit, Beth. Let me go back in history a little bit. You know, back in the day, when I started a long time ago, we used to use two different disinfectants. And a lot of time it was either a quaternary ammonium and a phenolic in some sort of a rotation, or a higher pH phenolic and a lower pH phenolic. And what we found as an industry is that residues left behind by the previous disinfectant, if they weren't properly removed, could adversely impact the ability of the next disinfectant to function properly. So the idea of rotating disinfectants, which was that theory years ago, is no longer in use today. Today, what we typically do is we qualify more than one disinfectant, because while there's no scientific evidence to say that microorganisms will develop resistance to the disinfectants that we use in the process, unlike antibiotics, which is a known phenomenon, disinfectants are much, much more powerful than antibiotics are in killing microorganisms. So anyway, but we would still qualify two different disinfectants just in case we had a supply chain problem, or even if the flora changed, right? Sometimes disinfectants are naturally resistant to one type of disinfectant when compared to another. But back to the question about the rotation, what we're really talking about is ensuring that one of those disinfectants that we're using regularly is a sporicidal disinfectant. And the reason why we do that is because the spore in its more resistant form is much harder to kill. So therefore, we want to rotate in a sporicidal disinfectant to ensure that our spore population is kept under control, or quite honestly, mostly eliminated. And then our routine disinfectant is used on a regular basis.
So I heard you say something really interesting there, Bob. Earlier on, I mentioned that I hadn't really considered purchasing or as being part of this, but you mentioned supply chain and the potential for the inability to get the products. And I think now more than ever, in the circumstances that we're dealing with, that could be critical and crucial. I mean, what do you do if you can't get the item or the sporicide or the material that you need to clean the facility with, or that qualifies?
Exactly. And that's why it's important that you have more than one qualified, so that you have, backup options. And obviously, you work to ensure that you have a sufficient stock to ensure that you're able to continue disinfecting on your defined schedule.
Yeah,
Right.
yeah, okay. All right, next question. Could you please talk about the cleaning requirements and steps for upgrading the clean room after refurbishment?
I'll take that one, Beth. So after there's been a construction activity in the space, we're likely to have a lot of contaminants that are not typical to our space, right? There may be some construction debris around, there may be some materials, whether that be small pieces of metal shaving or small pieces of wall material. And so it's really important to do a thorough cleaning first. One of the reasons why we talk about cleaning and disinfection is the fact that you can't disinfect through schmutz. So
Exactly.
if you have schmutz in the facility, that's a technical term, by the way.
I was going to say, is that a technical term, Becky?
You've got to get that out before you disinfect that facility. And so typically what most people do is have several days of very aggressive cleaning and recovery of that environment. They'll do some disinfection leading up to the start of that activity. And then, as we discussed, they might do another EMPQ at that time to check the performance and the control over that facility prior to resuming operations within that space. And so that EMPQ is going to be a time where we're monitoring the environment for several different parameters, both if it's a classified environment, we'll be looking at particulate non-viable as well as the viable that we might be detecting in the area. And we're going to want to get those measurements and ensure that we've returned to our original area classification before we resume any sort of manufacturing in that space. I don't know, Bob, if you had any other thoughts as I was talking?
Yeah, I just want to add on to that because I'm keeping an eye on some of the questions as they're coming in, and there was a question that you started to answer. I wouldn't say inadvertently, but I just wanted to add on to give a complete answer. So the question was about you're talking about cleaning and disinfecting, that they're two different actions. So cleaning with the disinfectant is not the correct way. So I think you started to touch on that. When it's very, very dirty, obviously we use a surfactant, right, to clean first, but the principles are the same, which is why we talk about cleaning and disinfecting in the same way. Unidirectional overlap strokes, make sure that you're rinsing your tools, whether it's a mop or a wipe, regularly to ensure that you're removing the soil. But what I think people need to understand is that, let's say in a controlled aseptic manufacturing process in a grade A space, there's not a lot of cleaning per se that I'm going to be doing on a wall, right? There is no soil that's being
under
generated.
Normal circumstances, not under construction, right?
No, exactly, under normal circumstances. Yeah, so therefore, we don't necessarily need to go in and clean. Many of the disinfectants that we have very, very small amounts of surfactant in them, specifically to just ensure that if there's a little incidental contact with a body or a cart or something, that we can remove minimal amounts of surface oils while we're doing the disinfecting. But that's why we tend to talk about cleaning and disinfecting in the same thoughts or in the same vein, but they are two totally different activities.
Right. And if we got schmutz, we got to take care of that first.
Absolutely.
If we don't have schmutz, then we can do it in one step.
If we're schmutz free, we're good.
That's great, guys. Thanks. And thanks for picking up that other question, Bob, as it relates to what we were just talking about. Okay. What would you recommend using on glass walls to avoid streaks and disinfect effectively?
Let me take this one, Becky, because I have a funny story about this. So back in the day when we used toxic chemicals to humans like glutaraldehyde, right, which we used to use on the regular, it would leave a horrible residue behind, right? And it was just aesthetically not pleasing. I had a boss at the time who insisted on seeing that residue left behind as a way to ensure that the operators were actually doing cleaning and disinfection. So again, we're talking about way back when.
You could tell coverage by how much stuff they left behind.
Exactly. But to answer the question, what a lot of people do today, a lot of our disinfectants leave very, very little residue. Hydrogen peroxide leaves no residue, as an example. But for those that do leave residue, oftentimes after there's a sufficient dwell, we might use, depending upon the area and depending upon the practice, we might use a sterile water rinse, a sterile WFI rinse to remove residue. We might use a sterile lint-free alcohol wipe on that surface to remove the visible residue and the visible streaking. So hopefully that answers the question.
All right, I love that story.
Probably why I have no hair.
All right, something about sporicide. When do you use sporicide?
Yeah, I can take that, Beth. So, you know, again, some companies will have a schedule and say it's time to use my sporicide and will respond by using it. But increasingly these days, we're finding that folks are only using that when they see that they're increasing spore counts in their area, or when their history tells them that it will be appropriate. And so if I've got this baseline bioburden in the background and I understand, you know, that I'll start to see spores during a particular season, or after a certain period of time without using a sporicide, then it's time to rotate that sporicide in. Again, we want to look at the materials of construction of our environment and let our environment direct where our program should go.
Right. Let me just add on to that. The reason why we don't want to use a sporicide all the time, Beth, is because it tends to be chemically aggressive against the surfaces, even against things like stainless steel. So it's a very harsh treatment compared in general to a regular disinfectant. So we try and limit it. But there are also times where you have a lot of material traffic, let's say at an entry material airlock, which you may use a sporicide, especially in something like the floors on a regular basis, as a way to control that contamination coming in. I think Becky's talking about it holistically, but there are certain situations where we would use it, you know, daily, let's say.
Yeah. Yep.
Okay. Yeah, here's another one about sporicide. So Compendia tells us to use sporicide on a very less frequency. This is in alignment with what we're talking about here. But nowadays, most of the disinfectants are sporicidal. How do we overcome that?
I would say not all disinfectants are sporicidal. There are certainly sporicidal disinfectants, and hydrogen peroxide at the right concentration could be a very common one now is hydrogen peroxide in combination with peracetic acid. So those clearly are sporicidal agents. But to the best of my knowledge, things like the quaternary ammoniums, the phenolics, those are still not rated to be true sporicidal disinfectants. They may have some efficacy against certain spores. Fungal spores are easier to kill than bacterial spores, so it's certainly possible that they will be efficacious against certain spores, but to truly be a sporicidal disinfectant and kill bacterial spores requires, special chemicals.
I agree.
Okay, all right. We've got a question here about the VR and about virtual reality and using virtual reality in a training program. So how would you use VR, like what was demonstrated in your training program? And I usually don't take questions, and Q&A is not really my, on these topics aren't usually my jam, but if you don't mind, I may take this question because of the experience that I've had with implementing Virtuosi and how other organizations are using it. You guys good with that?
That'll give us a break.
Absolutely.
Yeah, that's fine.
Take a breather, take a drink. So this question is about how do you use VR in a training program? So based on the experience that we've had so far with implementing Virtuosi into manufacturing sites, is that it's really used as a complement to your OJT and to your SOPs. And we've seen organizations have great success in minimizing facility time, number one. A lot of the places that we've been don't have a dedicated space for people to train on these types of things, on facility cleaning and disinfecting, so they actually have to use the manufacturing facility. So by getting the training in a virtual environment, you don't take up time in the actual manufacturing facility. And the other the way that we've seen benefits to what's been to implementing this VR is a SME time. I mean, we all know that the SMEs, and for those of you on the call that are SMEs, you are the most coveted people in the organization and are constantly called on for everything within your scope, and you're just pulled in a lot of different directions. And this has, we've seen this happen where it's eased up on the SME time because you are in the VR, you're getting trained without the presence and without the time of an SME. So the way that I've seen, like, to really circle back around to what the question that was being asked about how you incorporate it, is that it is a complement to your current training program in a lot of cases, and in at least one case that I've seen, it's actually replaced a big portion of the training program at the facility. So I hope that answers your question. I don't know if Becky, Bob, you want to add anything to that?
Yeah, I wanted to add something to that. We only saw a really short clip of the VR, and I think one of the things that the VR really is good at is teaching technique or basic techniques. And so, we saw in the clip there that there's a 3-bucket system. And so one of the things that VR excels at is ensuring that people are using the buckets in the correct sequence, right? And it provides a coaching alarm to point out when they've used the wrong solution at the wrong time. It gives them coaching on things like overlapping strokes, right? So when I'm doing the wall, it ensures that I've got the right overlap between my strokes. It ensures I only go top to bottom. It ensures that I don't miss any locations. And all of those alarms or coaching are in a very non-threatening way because it's a computer telling you, not someone looking over your shoulder. And so one of the things that it really helps with new people who are new to the task is really to provide them that coaching in an individualized way so they can do it as many times as they need to until they feel confident in that skill, until they understand why behind what they're doing with the system. And then that enables them to go to that on the job training with an SME, or to that real world training in a room with their equipment in a much more confident fashion. And so I think that's really some of the benefit and what that VR can teach us.
Yeah, thanks for adding that on. That is critical, like, to be able to make mistakes and learn from those mistakes. That's how we learn, right? In all aspects of our life. And if we're constantly being told, do it this way, and we do it that way, and we do it right, oftentimes we don't know why we've done it right. We're just doing what somebody told us to do. And that gets us in trouble down the road. That's going to get you to the next step of whatever training or whatever activity you're doing. But down the road, that's going to get us in trouble. There was something else, Becky, that you mentioned about the SME, like the coaching that is in the VR. We've heard from organizations, and this is solving that problem a little bit, is that a lot of times you have several people that you're training at once, and you can't keep an eye on everybody at all times. And this allows you to have one-on-one coaching when when your trainer or your SME may have looked the other way and is talking with somebody else about something, they miss something major that you may have done wrong. And within the VR and within Virtuosi, you don't experience that. So,
Absolutely.
All right,
And a lot of people these days have contract cleaners, and I see a question popped up about how do you qualify contract cleaners. One of the biggest challenges is that those people are generally, their turnover is more rapid than there might be otherwise because they are from an outside agency. They're not employees of your firm. You can't control necessarily who is showing up on a daily, weekly, monthly basis for your facility. So it's really important to train those people effectively and to ensure that they understand the why behind what they're doing. As we've said over and over, people who understand the why do things more consistently because they understand the importance of the procedures that they're following. They understand the requirements that they're trying to fulfill. And so that will enable us to, teach them technique first, teach them the why, then teach them the specific SOP, then expose them to the specific environment that we have. And that's so important for contract personnel, especially.
Yeah, and just to add on, I don't want to get on my soapbox here, but cleaning and disinfection of a facility, and again, going back to what the question posed, truly disinfection of the classified spaces is a critical task, right? Because it can directly impact the product that we're manufacturing, which then could harm the patient. So This idea of outsourcing such a critical task has always boggled my mind. I'm off
Right.
my soapbox now.
Thank you, Bob. All right, let's move on here then. Under what circumstances should fumigation or fogging be used?
Ooh, that's a good one. So I think that you have to use it very sporadically and very thoughtfully and carefully. If you're fumigating, it tends to indicate that you've got a perceived problem that is not located on the surface. So the question is, where is the problem coming from? And the environmental monitoring program, interestingly enough, gives you a snapshot of what the environment is. The cleaning and disinfection program, we assume, as we discussed before, works properly because we take environmental monitoring samples. So if I have to fumigate a space, it tends to indicate that there's a problem that I don't believe that my routine sanitization disinfection program is capable of handling. So it's a really tough question to answer, when and why, because of the likely root cause is that somehow organisms are getting in on a routine basis if I'm constantly getting these hits.
And I have seen some facilities use it on the times when they're restarting a facility after that construction event, for sure. And that kind of high efficiency disinfection of the area is really important in those cases because we have had so much external influence on the environment. But I would agree, in a well-controlled facility, it shouldn't be necessary. Now, if we talk about that kind of fogging within an isolator or a glove box environment, that's being done for a different purpose. That's being done because of the complexity of the surfaces and the difficulty in accessing all surfaces, because my only access is through glove ports. I'm not conventionally putting the man in the room or the woman in the room. I want to be inclusive to perform the task. And so there's a need to use that kind of intricate access that a fog or a disinfectant, vapor phase disinfectant can provide.
All right, great. Okay, there's so many great questions coming in here. Let's move on to another one. Where can industry stakeholders find the best definitions of the terms cleaning, disinfection, sanitization, sterilization, as they relate to surfaces, agent chemistry, and viable organisms and spores, as these terms are often incorrectly used?
Yeah. Is it PDA TR 71, Becky, that talks about the cleaning and disinfection program?
I think so. You're stressing my number recall at the moment.
But I remember it was a more recent one that came out probably within the last 5 years or so. My recollection is that had some pretty good definitions in it.
There's also a book by Seymour Block. I actually think it's sitting right next to me.
Yes, Dr. Block.
Of course it is, Becky.
Here it is.
It's the Bible.
Disinfection, sterilization, and preservation.
Yes.
And this is a very big book. And I wasn't using this in reference to this course. It was just sitting next to me as a matter of course. I almost
prop
forgot.
your monitor up on for the webinar.
This is the 5th edition, but I think there's actually a 6th edition, possibly even a 7th now. But this is the Bible of disinfection and talking about the topics of what they mean and what agents can be used and how they work.
Right.
And so now that you've seen what a geek I am at home, we'll go back to the regular questions.
That's great though, Becky. I mean, it's good to know, like, look, even our experts, even the industry experts are using these references to support the work that they're doing.
Absolutely.
That's important to see.
Absolutely.
Yeah. Okay, how many times should we clean and disinfect before upgrading?
Before upgrading.
Oh, I think, and again, we'll have to take a little guess on this, but Becky, I think this relates to your coming out of a shutdown and maybe clean and disinfect before you put the area back into service or consider it a classified environment. That is a question that you would have to answer based upon what disinfectants you use. But typically, we see people do it 3 times, and there's no magic behind 3. I've heard a variety of answers, but by doing it multiple times in a row, what you're able to do is during your this in situ study, as Becky was talking about, is that you can see the contamination fall at a certain rate by disinfecting, sampling, disinfecting, sampling, disinfecting, sampling, and then that study sets you up to say, do I need to do it once? Do I need to do it twice? Do I need to do it 3 times? And then coming out of a shutdown, provided that it's as that your in situ study was done under such a worst case condition, you can use that data to say, how many times do I have to clean before I restart my facility? And again, you might restart it at risk until you have routine environmental monitoring data to say that my environment was in a state of control.
Yeah, and ultimately your data is going to tell you in the end that you were successful in that endeavor. And over time, your history is going to inform you whether that 3 times a charm rule actually works effectively.
I hope it
Just
does in my
remember,
case.
it's only sampling, right? We're not covering every square inch of our environment with a sample over time. And so we're really counting on some probabilistic determination that we are clean. And so this is one of the reasons why we do things multiple times, because it will help us gain confidence in the robustness of that condition.
Okay. Here's a really good one. How do you use IPA wipes on a surface using overlapping strokes? For example, do you fold the wipe after every use, 4-fold? How do you do that?
Becky, I'm having a little
a little flashback
here. Drawing those pictures.
So we have an excellent section on this in Virtuosi, and I'm sorry that we didn't think of it for this particular activity, but we do talk about how to use a wipe in the activity, and we talk about how you can fold a wipe and that you need to use each side of the wipe only 1 time so you don't recontaminate any surfaces that you've had. And at the end of this very complex demonstration of how you can do it with a folded wipe, we point out that it is far easier to use a single wipe, use it for 1 stroke and throw it out, and not have to deal with folding it. Because every time you fold the wipe, you end up with a risk of touching the surface with your gloves, which would contaminate you, not only the environment that you're about to sample. And so we do not have stock in the wipe manufacturers of the world. However, it is far simpler to use a single wipe and single stroke again. Now, as we've worked with Virtuosi with various customers, we've had a few say to us, hey, when I'm starting up my biosafety cabinet, I am allowed to use the same wipe for 4 strokes instead of just a single stroke. Am I doing it wrong, right? And the answer is, well, a single stroke would be a much more conservative approach for using that surface, and is certainly the safe approach. If I'm cleaning, as Bob said, or disinfecting a clean surface that already has no schmutz and also was sanitized last before I got out of the previous manufacturing activity, and now I'm going back in and sanitizing again, which is already a sanitary surface, I have very low risk if I choose to use a wipe more than 1 stroke. But you know, from a technical scientific standpoint, we really would want to preach a single stroke, 1 and done for the cloth that we use.
Yeah, it sounds like there's a practicality to it by doing it singly and not having to worry about fumbling around. So I'm going to pause us right here. I know we're running close to the top of the hour, and we do have another half hour of Q&A, another 30 minutes, but I know many of you may only be able to stay for the hour. So I want to make a couple of quick announcements, and then we will move back into the Q&A. So today is the second of our 4 session Thought Leader Thursdays. So we have 2 more coming up next week. On November 12th, we have bacterial endotoxin testing, and on November 19th, we have manufacturing equipment use cycle. So keep an eye out for those. You can see us on LinkedIn. You can find us on LinkedIn. If you're not getting an invitation, certainly look for us there. If we have not answered your question, we will get back to you. If you have to jump off, we will get back to you if we were unable to answer it. Or if you have a question that you weren't able to ask, you can ask those questions or send those questions into us at info@virtuosivr.com. And we will get those and we will respond to you with an answer. And to learn more about Virtuosi, visit us at virtuosivr.com or reach out to us at any time at that same email, info@virtuosivr.com. Okay, so hopefully you all are able to continue to join us as we continue on with the question and answers. We still have lots of questions to respond to, so let's get right back into it. All right, can we use sporicide on gloves, or is there any other sporicides that are safe to use on gloves during aseptic operations?
Let me start out, Becky, and then you can pile on afterwards. Let me start out by saying that when we are putting on sterile gloves during our gowning process, and if we're following proper aseptic technique, there should not be spores on the gloves, right? The gloves are sterile when we're putting them on. We put them on with a technique that doesn't contaminate them. We're handling sterilized equipment. We're in an environment that presumably grade A, grade B environment. That should be very, 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 very low bioburden. So therefore, if I'm getting spores on my gloves, I've got a different problem than using sporicide on my gloves. I have to figure out where those spores are coming from, how they're coming in, and if I was a betting man, most likely I would say that it's on some sort of piece of equipment that can't go through the autoclave, maybe an environmental monitoring piece of equipment, maybe a cart. But it's really more important not to think about using a sporicidal disinfectant on gloves, but really to understand how spores are getting into that area and eliminate that.
And how my people are coming in contact with them, because that means my people are touching those surfaces as well, and that is a high risk. So I absolutely agree with you on that.
Okay, all right, great. Next question here. Can you, let's see. How would you recommend dealing with a bioburden organism spore former such as a
Pennibacillus?
Thank you. That is not being eliminated with a general sporicide. Yes, Becky comes in for the win. Thank you. Thanks.
So one of the things when Bob was talking about the laboratory studies, and he mentioned that we test the disinfectants that we use in the laboratory first. One of the things that we do is we typically include environmental isolates as part of the panel that we test with. And the purpose of doing disinfectant efficacy with an environmental isolate is to ensure that when we have something that is resistant to that particular organism, that we're actually ensuring that we've got the right chemistry for that particular species. And you know, if we are demonstrating routinely in the lab that the organism that we're concerned about is being taken care of with that chemistry, then we need to look to something like technique, right? And one of the things that we look at in the lab is contact time, and we define that as wet contact time. And that's the time period in which the solution must remain on the surface in a wet state to conquer whatever the organisms are. And of course, spore formers are much more resistant than other types of bacteria that we encounter in our environment. And so it's important that they be exposed to the chemistry for the designated period of time. It is essential then that we ensure that our surfaces are remaining wet for that same wet contact time in the environment that we experienced in the laboratory. And one of the things that we'll see is that in clean rooms, the relative humidity is low, the airflow is high, and one of the things we'll see is that surfaces are drying faster than we experienced in the laboratory. So it really needs a careful eye to determine whether a solution should be applied to the same surface multiple times to achieve that wet contact time. So let's take an example. if in the laboratory I determine that something took 10 minutes of exposure, I need to make sure that that surface stays wet for the entire 10 minutes. If I start to see evaporation after 3 or 5 minutes, I might have to apply that solution 2, maybe even 3 times during that disinfection time period to ensure that I achieve that duration that I've proven in the laboratory is effective. And so, you know, if I see something that's not being eliminated with a general sporicide, I go back to check what my lab studies had shown me, whether I tested that isolate. I might do a test specifically for that isolate, and then I want to look at those techniques that I'm employing in the real world. Bob?
I think you hit it. I think you were very comprehensive. I would say the only other thing to consider is if you're confident that your chemistry is working on the surfaces, and if you're confident that you've got the wet contact time in the environment, yet you're still having recoveries, then it may not be the disinfection process. You may be reintroducing this organism from somewhere. So you might have to look at either an engineering solution or a process control solution.
Right. Right.
Okay. All right, great. Let's see. What period of time should the cleaning PQ cover? For example, 3, 6, 12 months?
So the interesting thing about that is, you know, we talk about a PQ. We start out with a dynamic sampling typically. A static sampling typically, and we want to do that to ensure that we have the ability to start with a clean environment, right? We don't want to jump into a PQ and find out that we didn't adequately disinfect the environment to get it to the level that it needs to be, because obviously we do a PQ in a controlled state. So we usually do some days of static sampling. I've seen people do, you know, 3 days of static sampling just to say, yep, I'm confident that I'm starting at my criteria or below, you know, my criteria. And different rooms, ISO 5, ISO 7, ISO 8, of course, have different criteria. So we really want to make sure that we're at that criteria. Then we start what we call our dynamic sampling, or sampling when normal operations are occurring. And this can either be a very structured activity for a couple of days where I try and put the maximum number of people in the room that I would normally have, as a way to demonstrate what load my room can handle and my disinfection process can handle, and still maintain that room in an environmentally controlled state. Now, a minute ago, I mentioned to you about engineering controls. This is where room air changes is a big part of it, right? As humans are in the room, even when we're fully gowned, there's the potential of shedding skin. It's important that I have enough room air changes coming down from HEPA filtered air, which I know is clean, and flushing out of the rooms to the low wall returns to really remove those microorganisms from the room before they get a chance to settle, before they get a chance to proliferate. So now that we're talking about the process, you know, now we talk about the time, right? The time interval. At that point, it's really just a matter of how long do you want to keep the protocol open so that you can demonstrate a state of control under normal conditions. Now, the interesting thing about the environment is that we typically have already built into our programs a requirement to report trends and analyses, usually on a quarterly basis. And in many times we summarize it on a yearly basis as a way to see year-over-year trends or year-over-year activities. We talked before about some seasonal changes that can affect the flora in the environment. I remember one place that I was at, Bacillus thuringiensis used to come out every fall, and I forget why. There was a reason. It had something to do with leaves. Becky, you're more about you know, the micro than I am, but that came out every year. You didn't see it, you know, for the first 9 months, but boy, in September, October, you'd see a spike in this particular organism. So anyway, the point is that you're already kind of trending that information for an extended period of time. So it's really just more important when you define the end of the quote unquote PQ, whether it's, you know, a week, 10 days, 14 days, a month, to analyze that chunk of data and report on that chunk of data. And then you're really analyzing it, reporting it over a longer period of time anyway.
Right. That data becomes the reference point, the touch point to what you say is a good state of control, because you should have a good state of control when your facility's first starting up in that environment. And then that becomes a touchstone for the rest of the data that you collect. I would say one of the other things, Bob, you mentioned doing dynamic testing when there's activities in the environment. It might take you a given period of time to cycle through the different
Sure.
types of activities you have, right? And it's
Mhm.
really important to simulate all of the different high flow activities, the high interaction activities that
Mhm.
you might have in that space.
Right.
And you know, unless you've created some sort of simulated worst case activity that you're going to do, you're going to want to go through a normal range of operations. And so that again
Absolutely.
should inform how long that is.
Mhm. Yeah, you know, Becky, let me just add on to that because you know your point is very well taken, and especially now that we're seeing a lot of facilities that deal with disposables, there's a lot more materials that are being brought into the environment than what used to be when everything you know was fixed equipment and whatnot. So that load, and to your point, Becky, that you know the time I may only bring things in once every 21 days depending upon my process, so I would have to keep my PQ open, you know, for that period of time unless I could simulate all of those things. Excellent, excellent point.
Yep.
Great, thanks for that. That was great discussion. And hopefully that answered the question for the participant. Okay, is there a guidance document on establishing a cleaning plan? Becky, do you have one just lying around that you can hold up? A guidance document?
I did not. I've
Oh, Bob, there
I've
you
run
go.
out of things in my kitchen that I can tell you.
I'll be honest. I don't know that there's.
Virtuosi.
Well, yeah, but this is true. That is a guidance information, right? Virtuosi does a good job of that. But I can't think of anything in regulatory guidance that I've ever seen about a cleaning plan.
No, I haven't either. And I think that's one of the things that, if I can add a plug, that's unique about what you did. And Bob was actually one of the authors for our cleaning and disinfection section in Virtuosi. And one of the unique things that he did is he brought it to that practical level of what do I do when I'm faced with a room with a bunch of tanks in it and control panels and this other equipment? How do I really have to set up my standard operating procedure? And that's really what it's about, is making this science come down to the level of practicality.
Yeah.
Okay, great.
So
Yeah,
my answer would be Virtuosi.
Thank you for the plug, Becky. Okay, so many great questions coming in. You spoke a lot about sporicide. Can you talk about other cleaners and disinfectants and when, why, where they would be used?
Sure. You
Yeah,
want to start out, Becky, or you want me?
I don't care.
So you know, we talk about let's separate them because I think you know we talk about them together, but let's talk about cleaner. So cleaner is a surfactant that in and of itself doesn't necessarily have microbiocidal activity, although you will see some microbial reduction just from the surfactant action of washing things away. I mean, let's think about washing hands. We're using soap, right? So that surfactant allows us to get things off, and then when we use the water, it rinses things away. So you'll get some microbial reduction, but not necessarily through biocidal kill. Okay, so I would only use cleaners in areas where I have soils or where I'm generating soil. So certainly in my entry airlocks, depending upon my process, if I've got carts and wheels and hopefully not pallets or drums on rolling wheels, but those are the areas where I might have to deal with soil. So in those areas, I would start with a surfactant. I might also start with surfactants on things that are wrapped. That are coming into the facility that may have been packaged in a cardboard box, for example. I would take them out of the cardboard box at a place distal to where my airlock is, but then before I introduce them onto, let's say, a transfer cart, I would use some sort of a cleaner to ensure that I've removed any type of residue or dust that might be on the outside of it before I start my disinfection process. So that's pretty much the cleaners. As far as disinfectants go, I've mentioned a couple different chemistries. Phenolics is one of them, and I know that they have higher pH phenolics, lower pH phenolics. We have ammonium compounds, quaternary ammonium compounds that are typically classified as disinfectants. Trying to think of some other chemistries. Becky,
IPA.
Yeah, sure. I always forget about that. Alcohols, right, or disinfectants. I think we've all learned, how to use hand sanitizers and whatnot. But they have biocidal activity, and we use those on a frequency commensurate with the area. And I think early on we talked about, based upon the environmental grade and based upon data that we've actually collected. So typically my most critical areas I'm going to disinfect every day because my limit tends to be 0, or in a grade B space, ISO 7. It tends to approach 0. So I would do that absolutely on an almost daily basis when it's in operation. For my lower grades, my ISO 8 areas, let's say, or my C and D areas, there I might disinfect less frequently, but I would also look at the activity. So going back to my airlock example, that entry airlock might be my lowest grade classification, but it's going to be my dirtiest area, right? So I don't just want to look at it and say, it's a low grade, I won't disinfect often. It's what process is going on in that room. And because the process is dirty, relatively speaking, I want to disinfect every day, maybe even multiple times a day, to ensure that these contamination doesn't get into my facility, right? I have to, I literally have to stop it at the door, as it were. Talk to the hand.
Yeah. And then of course, as we've been talking about, the highest level is that sporicide, which I use intermittently as dictated by my flora and my facility.
Right.
Okay, great. Thanks for that thorough response. All right, next question. You guys doing okay? We've been answering questions for a while. You got a few more in you?
Coffee.
You do this all day.
All right. Well, I know you guys are passionate about the topic, so I figured as much. Is there ever a time that we would validate the environmental cleaning program as part of the cleaning validation process?
Yeah, so let me take that, since cleaning validation happens to be my passion, right? So generally the answer is no. So generally when we talk about cleaning validation, what we're talking about is cleaning product contact equipment, right? So this is the equipment that is going to touch the material that's ultimately going to be given to patients. And when we talk about environmental sanitization and disinfection, we're talking about the room that surrounds that particular space. And so we typically separate the two things, and there's a validation for the patient risk surfaces, and there's a qualification for the environment, but it's not at the same level. We're not analytically testing for how much a disinfectant is left over after the cleaning process or the disinfection process is completed. We're only testing to see, have we achieved our microbial goals? Whereas on the equipment, we're looking for product residue, we're looking for detergent residue, as well as the microbial contamination. Now, in some facilities, if they're working with high potency products, if they're working with something like a steroid, for example, or something hazardous that's going to be very harmful to the operators as well as to patients, should it be in the environment or should it move around our facility, we may actually sample for that particular product as well in the environment to ensure that we have good contamination controls. And so contamination control is part of what we want to evaluate with regard to both our engineering controls and our cleaning programs in that environment. When we, interestingly enough though, when we shrink the room from walls, floors, ceilings, and bring it down to, say, an isolator size space, we typically do include some of those isolator surfaces in our cleaning validation program. And the reason why is because they've increased their risk level to our process because of their proximity. And so there may actually be some sampling of those surfaces that gets included in our cleaning validation program, simply because although they are not direct product contact, they are potential contributors. They could shed into the environment based on their proximity. And so that sort of differentiates the different levels of testing that we might do in these environments.
Yeah, that's great. Thanks for that, Becky. Got so many great questions. All right, is it okay to use sporicides for disinfection directly before OQ, though we won't use that for regular disinfection?
So let me take an
I'm guessing to clean up
interpretation
the
of that.
environment, right?
Yeah, yeah. So that's why I'm saying I'll take a stab at trying to answer it. So there's a difference between what we use routinely as part of our disinfection program and getting my environment in a state of control to be able to start my environmental monitoring performance qualification. So assuming that this is what the question was, even though the question said OQ, if they meant EMPQ, then I would say the answer is yes, because the idea isn't to well, the idea is to ensure that your environment is below those levels that you've set for your environmental monitoring. So 5 CFU, 10 CFU per surface, whatever those limits are depending upon the environmental classification. So yes, you can use the sporicide. I would encourage you to consider routinely using a sporicide from time to time, although again, not every day as we've talked about, as part of that routine disinfection process over time, because I can almost guarantee that you're going to need it periodically. But did you interpret the question any differently, Becky?
No, I thought it was the same thing. Yeah, we
Okay.
can use the sporicides initially to get the facility under control. Because
Sure.
picture, for example, if you did construction where you had to dig up the floor or you were putting up new walls, there's a bigger opportunity for spore formers to enter the facility when you're bringing in wood materials, when you're disturbing dirt, you know, any construction debris that they have. I mean, equipment as it's shipped to you might be shipped in a wooden pallet or case, right? And as that material is removed from the clean room, it's not just about sweeping up the wood chips, right? It's about sweeping up the stuff that's left behind from that handling or from that exposure. And so a sporicide is a very appropriate agent to use in that particular case to recover and get back to that good baseline that we have. So yeah, I took it as a startup question, Bob.
Okay.
Okay, great. All right, moving along. How do you qualify operators to visually inspect equipment to ensure it is clean?
So, okay, now we're straying into cleaning validation, which again, I could talk about all day.
Go for it.
So how do we qualify operators? It's usually a multi-tiered program, right? Of understanding, instructing them on what equipment should look like when it's in a state of control, teaching them how to use the tools that they're going to have. So they're going to have mirrors, they're going to have flashlights, they're going to have the naked eye in a room, and teaching them where to look and what to look for. I think even more importantly for most people's programs, though, it's not just about how to look at a piece of equipment, how far away from the surface to be, how much light to have on the surface, what angle of viewing you should use from the surface, but also about what to do when I see a spot, right? How do I determine if that spot is a water spot that's left over or product that's left over? And how do I determine what clean looks like, right? It's about establishing standards and having those standards for them to compare against and understand what they see. As people inspect equipment, they're going to come across a droplet of water somewhere, and that droplet could be minuscule. It could be the size of the head of a pin, but they have to know, how do I respond to that? Do I say, it's no good, throw it out, rewash the whole piece of equipment? Or are they permitted to wipe it away with an IPA wipe because of the small nature of that material? So the biggest part for me isn't just the instructions on the inspection, but the instructions on what to do when I see or suspect something on the surface. But really, that's cleaning validation and not the subject of today.
Exactly. Tune in for.
That's right.
The next webinar series, right? But thank you for fielding that question anyway.
Sure, no
we
problem.
Appreciate it. All right, let's move on to another one. We'll see how many more we have time for after this, but let's get to this question here. Is cleaning and disinfection of isolators analogous to cleaning and disinfection of a room?
So I think I just sort of answered that a moment ago when I talked about the walls of the room shrinking into the isolator walls. It's, you know, the isolator is an analog for our processing room, but it's smaller and closer to the process. And so it's a little bit higher risk for shedding back into those open containers or open products or processes. So in general, yeah, it's a analog to the room, but it's going to be have a little bit more scrutiny because of its proximity.
Right, and the other thing that I would add to that is, you know, first we clean, as Becky pointed out, and then we might even wipe down the interior surfaces with an IPA wipe to sanitize. But what makes isolators, I don't know if unique is the right word, but what we do with isolators that's different than a room is we do fog them, right? Using that terminology that someone said before, right? After we clean everything, after we place everything, after we wipe surfaces down to make sure that they're clean, then we actually do a decontamination process of the isolator, typically using vapor phase hydrogen peroxide.
Right.
Okay. All right, great. So I think we've got time for just one more question here. Let's take this one here. What is intended by the phrase contact time?
I talked about this earlier, Bob. Why don't you grab that one?
Yeah, so contact time, you know, is the amount of time that I need my disinfectant to be in the aqueous state, you know, liquid, in contact with the surface to ensure that I'm killing, that I have sufficient time then to kill the microorganisms. So if we want to equate it to cooking, right? Contact time obviously is time, but if I put a pizza into an oven at the correct temperature and I only leave it in there for a minute, it's probably not going to be done. Right? And if it's so hot that it is done in a minute, it's probably not going to be tasty because it's going to be charred, right? So I need to have that right chemistry, which we've talked about. You know, the concentration of the disinfectant, which is the temperature of my oven. Then I need my contact time. So I need that 7, 8, 10 minutes for it to bake properly, to come out with the perfect New York thin crust, bubbly cheese, and oh boy, I'm getting hungry.
Last time we do one of these before lunch.
Exactly.
Yeah, right.
But all kidding aside, that's what we mean by contact time. It's the amount of time that the surface needs to be in the condition, in this case in the wet condition, to ensure that I have the appropriate time to kill the microorganisms.
All right, great.
Well,
I
pizza
think
for lunch.
Yeah, I know I'm getting hungry now too. So I think that's going to wrap up our session for today. So this was really, really excellent information from both you, Becky and Bob. I really appreciate your time today. I can speak for the participants that we've all learned a lot, and it was a lot of fun. You know, facility cleaning and disinfecting is certainly something that you all are experts in and passionate about. So thank you for sharing that with us today. We've got 2 additional topics in the up and coming weeks for Thought Leader Thursdays. As I mentioned earlier, this is the second of 4 sessions. Next week, November 12th, we're going to be covering bacterial endotoxin testing. And on November 19th, we have manufacturing equipment use cycle. So keep an eye out for invites for those. You can find us on LinkedIn and register directly through LinkedIn. For those of you who are able to stay with us, thank you so much for joining us. You know, this Q&A session, I've mentioned it a couple times already, but this really is my favorite part about the webinar format that we use here at QXP, because Becky and Bob are truly industry leaders on these topics, and it's not very often that you get to just ask questions, whatever you want to know, to these industry experts. So we're so happy to be able to offer that to you. So thanks again for your time, everybody, today. Look forward to seeing you all on the webinars the next 2 Thursdays. Have a great day, everyone.
Great. Thank you, Beth. Thank you, Becky.
Yep. Bye-bye.
Bye.